Okay students, welcome to the new lecture, and in this lecture we are going to discuss about the new topics, that is growing roots. After this topic we are going to discuss about the tissue. Okay, we are going to discuss about the tissue, that is nothing but the plant tissue. And make sure if you have any doubts or questions or anything to know, please post it in the live chat section so that I can answer to it. Okay, so I am able to see the live chat section. Okay, good afternoon Geeta Shri, good afternoon Krantikar. Okay, good afternoon. So enter your doubts or questions in the live chat section so that I can answer to your questions or doubts. Okay, now coming back to the growing roots. Okay, so what is meant by growing roots? Growing roots are nothing but, okay, they are the roots of the plant. Okay, they are helpful in the absorption and so on. So next, Monica, good afternoon, good afternoon. So next, growing roots. These are the growing roots. So in the last experiment we have seen that the plants are going to grow the roots. Okay, and also we have done the experiment in onion. So what we have done in that experiment, we have taken the onion bulb, we have placed above the glass. So this is the glass. Okay, so we have filled the water in the glass and we have placed the onion bulb. Okay, so after placing, what you can see the growth of roots. You can see the growth of roots in the onion bulb. Beneath the onion bulb, where there is a touching of the water, there you can see growth of roots. So by this way you can see the growth of roots in the onion bulb. So let us remember, this is all completely, it is the water. Okay, okay, Hemashri, good afternoon. And if you have any doubts or questions, post it in the comment section. Okay, so next, in the onion bulb, beneath it you can see the roots, growth of the roots. So in this experiment our aim is to observe the growth of cut roots. We have to see whether the cut roots are able to grow or not. That is our aim. Okay, our aim is to check whether the cut roots are growing or not. That is our aim. So for this we need materials like onion bulb, permanent marker. Okay, next glass slide. So next to glass, not a glass slide, just glass. It is nothing but a cup with water and also scalpel. Okay, scalpel is nothing but a small blade. So here the first procedure is to take the onion that is used in the previous activity. In the previous activity we have used one onion bulb now. So we have to take that onion bulb and cut the roots. So with cut roots, already we have cut the roots of that onion bulb. So mark the roots where there is cut portion. Wherever there is a cut, so mark that portion and keep it onto the above the water cup. In this way you have to keep it. So here we have marked with marker, black colored marker. So wait for 4 to 5 days. And also check whether the roots are growing or not. Okay, wait for 4 to 5 days, and after 4 to 5 days check whether the roots, cut portion of the roots are growing or not. Okay, and make sure the level of the water is touching the roots or bottom of the onion bulb. So by this we have to make sure this. Okay, so in this way you can see. So observation, our observation is the cut roots of onion bulb did not show any growth. Here you can see the cut portion, whatever the cut portion is there over here, you can see there is no growth. Why there is no growth? That is the question. There is no growth. Why? Because here you can see the roots lost some tissue. Okay, so the roots lost some tissue. As a result, the loss of some tissue in the roots did not show any growth. So let us consider this is the growth. This is the root. If it is in this way, it is going to grow. But what we have done, we have cut the root. Okay, we have cut the root and we have marked with the black colored marker. So this is not going to show growth. This root will not show growth. Why? Because it has lost the tissue. Here the tissue, whatever the tissue is there over here, it is lost. Okay, as a result it is not going to show any growth. Okay, Amrinder, good afternoon. If you have any doubts you can ask me. Right, so here this root is going to grow. This root grows in the same way. Why? Because it has some important tissue in this region. This tissue is present. This tissue is going to help the roots to grow properly. Okay, so that is about our experiment. Okay, So based on this experiment, we found that there is growth of the tissue. Okay, the tissue is responsible at the tip of the root for the elongation of the roots. Okay, so the next result, our result is the cut portion of the root did not show or do not show any growth due to the removal of the particular arrangement of the cells at the root tip. At the root tip, some arrangement is removed, so we cannot see any growth. Okay. So next is cells in groups which are nearly similar in functions perform similar function is called tissue. So in the last class also we have discussed about the tissue. What is meant by tissue? Tissue is nothing but the group of cells. The tissue is nothing but the group of cells which has a similar size, similar function, that is known as tissue. Okay. So what is meant by tissue? The tissue meaning is the cells which have the same shape. Okay, see here, all the cells have the same shape. They have the same size. Okay, they have the same shape, same size, and they have the same function. In that condition, you can call them as a tissue. Okay, what is tissue? The group of cells which have the same size, same shape, and same function is known as tissue. Okay, this is the meaning of the tissue. So next is the types of plant tissue. In the plant, we know that the plant is multicellular. What is meant by multicellular? Multicellular means which has more than one cell is known as multicellular. Here we are saying that the meristematic tissue. The plant has different types of tissue. What are they? First type of the tissue is meristematic. Meristematic tissue is the first type. Second one is the dermal tissue. Okay. Third one is the ground tissue. Fourth one is the vascular tissue. So these are the types of tissue that you can see. Okay. So next, what is meant by meristematic tissue? Meristematic tissue means that these types of the tissue are involved in the growth and repair is known as meristematic tissue. So for example, let us consider there is a plant. Okay, there is a plant. So if the plant has some small damage over here, what is happening? So the damaged portion has to get repaired. So here, this type of tissue known as meristematic tissue is going to help in the growth and repair of the plant. So this tissue is present on the top sides and also in between layers of the tissue. Okay, that is known as meristematic tissue. So it is present both in roots, shoot tips, and also sides and so on. So this tissue, meristematic tissue, remember one thing, they have the thin cell walls. What is meant by thin cell wall? Thin means very, very light cell walls. Okay. So in the last class also we have discussed what is cell membrane. Cell membrane is a layer, layer around the cell. It is present in both animals and also plant tissue. So above the membrane, plasma membrane, there is another layer. This layer is known as a cell wall. Okay. So what they are saying, they have the thin cell wall. It means these types of tissue, meristematic tissue, has a thin cell wall. Okay. Next, they are cell wall with a prominent nucleus and cytoplasm. The nucleus, and also cytoplasm is clearly visible. Okay, that is in the meristematic tissue. So they are continuously dividing and involved in the growth and repair of the tissue. So just now we have discussed if there is any damage to the plant tissue in that region, this meristematic tissue is going to involve in the division and also repairing of the tissue. Okay, so this is about the tissue. Okay, so if you have any doubts, you can ask me in the live chat section. Okay, you may have the doubts or you may have the questions or whatever it may be. Do not forget to post your question or doubt in the live chat section so that I can answer to your question or doubt. Okay, so this is about the meristematic tissue, and here they are continuous dividing involved in the growth and repair of the plant tissue. Next is the tissue are compactly arranged without intercellular spaces. What is intercellular spaces? So this is one cell, another cell, and so on. So can you see between the cells there is space? No, here between the cells there is a space. Okay, the space between the cells is known as intercellular spaces. So shoot tip, it is not shoot tip. Yes, actually this meristematic tissue is present in shoot tips. Okay, lateral sides, etc. Okay, you are correct. So Ram Dular, you are correct. But so what we are discussing that intercellular spaces. Intercellular spaces means between the cells. Okay, between the cells there is some space that is known as intercellular spaces. Inter means in between. In between there is space. That space is known as intercellular spaces. Okay. So next is they are without intercellular space compactly arranged. This is normal. In normal cells you can see such kind of tissue. But in case of meristematic tissue, there is no intercellular spaces. They are compactly arranged. You can see here. There is no spaces. There is no spaces and these cells are compactly arranged. Okay. So this is known as compact. What is meant by compact? Compact means without any space. Okay, that is known as compact. Okay. So next is, okay, Pavitra, good afternoon, and if you have any doubts, ask me. And the next one is, based on the function and location, they are divided into apical, okay, apical, lateral, and also intercalary meristem. So this is apical, A-P-I-C-A-L, apical, okay. So apical, lateral, and also intercalary meristem, okay. So what is apical meristem? It means these apical meristem, lateral meristem, intercalary meristem, these all are the types of meristematic tissue, okay. So MT means meristematic tissue. Meristematic tissue can be divided into apical meristem, okay. Next it is divided into lateral meristem. Next it is divided into intercalary meristem. So these all are given over here. So apical meristem, okay. What is apical meristem? They are present at the top. Apical, what is the meaning of apical? Apical means on the top, okay. Apical means on the top. They are known as apical meristem. So and they brings about the growth and length. So on the top of the tissue or the top of the stem, these tissue are present. So next is lateral meristem. Lateral means on the sides of the plant, these tissue are present. So we are calling it as a lateral meristem, okay, lateral manner. So they are going to give rise to a growth and also girth of the stem. Girth means so size, okay, size of the stem. So intercalary meristem. Intercalary means between they are present, these tissue are present in areas of the branching and also stalk. So here you can see in this diagram we can clearly see this is apical meristem. This is apical meristem. So what is apical meristem? On the tip, can you see here there is a tip, okay. So on the tip the tissue is present. This is known as apical meristem. So next is the lateral. Lateral means on the sides. On the sides there is a tissue that is known as lateral meristem. So intercalary, what we have discussed, intercalary means in between the areas of branching, okay. Can you see here there is a branching, intercalary stem, there is a branching, so this is known as intercalary meristem, okay. So this is a thing that you can see clearly, okay. So lateral, apical, lateral, intercalary, these three types are the first type of the tissue that is known as meristematic tissue, okay, that is known as meristematic tissue. So next is composition of the meristematic tissue are root and shoot tips. So they are present more in the root tips and also shoot tips, okay. So if you carefully observe the figures of the root tip, okay, in this diagram you can clearly see the tip and also shoot tip, okay. Shoot means what? Branch. Root means what? In the ground there are roots, okay. In the roots and shoots there is the tips. In those tips these meristematic tissue is present, okay. And find out the differences in the diagram and also write in the table. There is a table, or write it. If you are unable to write, we will discuss in the next class, okay. So next is a Dicot stem. In that only we are going to discuss some more points, that is Dicot stem. So what is meant by Dicot stem? Okay, what is meant by Dicot stem? Dicot stem means the stem which is, okay, and if you have any doubts you can ask me in the comment section. I will give for 2 to 3 minutes, you can ask me doubts. Okay, what is meant by Dicot stem? Dicot stem is nothing but these are plants. These are the plants, okay, that have originated from a seed which has 2 cotyledons, okay. So already we have discussed in the previous classes, okay. Dicot stem means which have the seed, which is the plant which is arising from the seed of the 2 cotyledons, that is known as Dicot stem, okay. So next in this activity we have to discuss Okay, we have to discuss one most important thing, that is observation. Okay, you have to observe the main important thing. Okay, so next is aim. What is our aim? Our aim is to observe Dicot stem under the microscope. What is Dicot stem? Just now we have seen Dicot stem is nothing but, so these are the cells. Okay, these are the cells, these are the seeds that are having the two cotyledons, that is known as Dicot stem. Okay, so next is branching means, Ramdullah, branching means. So branching means is nothing but branches. Okay, branching means branches. Can you see here? Here in this diagram, you can see there is a branch. Okay, so there is a branch over here. There is a branch in the intercalary meristem is present between the branches, so that is known as branching. Okay, so this is about intercalary meristem. Okay, next is composition of meristematic tissue that we have discussed just now. The activity 7, that is Dicot stem tissue. Okay, Dicot stem, here we have to discuss about observation. First one is the aim. Our aim of this experiment is to observe Dicot stem. So see, first of all we have to discuss about what is dicot and what is monocot. Let us take the peanut or groundnut. Let us consider this the peanut. If you open the peanut, it has two seeds. It has two splits. Okay, this is peanut. Okay, you can see one cotyledon and also you can see another cotyledon. So this is dicot. This is dicot. Di means two. Di means two. Cot means cotyledon. Okay, cot means cotyledon. Okay, so this is two cotyledons are present. Then what about monocot? Opposite is monocot. Monocot means a seed which is single. For example, you can see wheat. Okay, you can see wheat. You cannot divide the wheat into two simple parts, just applying simple pressure. Like example, rice. Okay, rice is also a monocot. So you cannot separate it. So this is only one seed. Hence this is known as monocot. Okay, this is known as monocot. Okay, if you observe, if you understood this, okay, if you have doubts in the monocot and dicot, you can ask now. Okay. So next is apparatus. Apparatus is Dicot stem, slide, cover slip, water, scalpel. Okay, these are the things that are needed or that are necessary. Okay, so these are the things that are necessary for this experiment. Materials, you can call the apparatus as materials also. So next is the procedure. So in this procedure we have to prepare a temporary mount. Temporary mount means temporary slide. We have to make a temporary slide. Okay, this is a temporary slide. And also TS. TS means transverse section. Transverse section means what? Let us consider I have taken the stem in this way. Okay, I have taken the stem. Okay, I have to cut. If I cut in this way, this is known as transverse. If I cut in this way, this is longitudinal. So what we have to prepare? We have to prepare transverse section of the stem. It means we have to cut in this way. As a result, I will get the tissue as a disc-like type. Okay, I will get the tissue in this way. Okay, I have taken this portion. Okay, so this is the portion I have got. So next is I have to place it on the microscopic slide. I have to place this slide, a small transverse section of the stem on the slide. So this is a tissue which is placed on the slide. So then I have to add a drop of the water. Okay, then I have to cover it with the cover slip. So then I have to keep it under the microscope and also I have to observe it. If I observe, I will get the clear diagram. Okay, I can see clear diagram under the microscope. Okay, so next, by observing under the microscope, compare with this diagram. Okay, you can observe the diagram under the microscope and also second diagram is in the textbook. So observe the diagram and compare both the diagrams. And also draw the diagram in your notebook. So observation, what you can observe over here? You can observe that the tissue observed under the microscopes looks similar to the textbook diagram. It means the diagram which is observed under the microscope and also diagram which is observed in the textbook. So these two diagrams are similar. Okay, these two diagrams are similar. So hence, based on this, we can confirm that. So these are the Dicot stems that most of the stems looks almost similar. Okay. Okay, Vaishnavi, good afternoon. And so next is result. Result is the TS, the transverse section of dicot stem observed under the microscope. This is our result. What we have concluded? We have to conclude that we have observed the dicot stem under the microscope. Okay. So next is the dermal tissue. What is dermal tissue? Dermal, the term dermal refers to the layer. Okay, dermal refers to the layer. So the tissue means the similar cells. So the cells, the similar type of cells, similar type, shape, structure that is seen on the layers of the plant is known as dermal tissue. Okay, so they are single. These cells are single, and they are a single layer of cells with a thick cell wall. Okay, they are with a thick cell wall. What is meant by thick? So in the meristematic tissue, we have seen the cell wall is thin. Okay, now in this case, the dermal cells. So this is meristematic tissue. MT means meristematic tissue. Okay. So now we are comparing with dermal cells. So dermal tissue are very thick. Their cell wall is very thick in this way. So compare the thickness. So this is very thick, and this is very less thickness. Okay. The thickness is very less over here in the meristematic tissue, but in the dermal tissue the thickness is more. Okay. This is only the difference between that you can see in the meristematic tissue and also dermal tissue. So next is they are going to protect the plants from the loss of water. So dermal tissue, remember one thing, they are involved in the protection against water loss. So we know that in the plant body the water is most important. The water has to be saved in the plant. So this is the reason to protect from the loss of water. This dermal tissue is used. Okay, it will prevent loss of water from the leaves, loss of water from the branches, loss of water from the other parts of the plant. So in the desert plants also, these desert plants, what are desert plants? Desert plants are nothing but the plants which grow in the desert. Okay, that grow in the desert. We are going to call it, we can call it as a xerophytes. Okay, XEROPHYTES. They are also known as xerophytes. Xerophyte means these are the plants that will live in the less water condition or less water area. Okay. Example, opentia, cactus. Okay, these are the two types of xerophytic plants, or you can call them as xerophytic plants. So in these xerophytic plants or in these desert plants, the dermal tissue is more and they are more thick. The dermal tissue is more thick and this dermal tissue is waxy. Okay, waxy means they have the waxy coating on it. As a result, the dermal tissue is important in this way. So this dermal tissue is also going to provide mechanical damage, protect from mechanical damage. Mechanical damage means the wind. Okay, so due to wind. Okay, Okay, or due to stress. Okay, due to stress and so on. So from these mechanical damages or due to some animals. So from these mechanical damages, this dermal tissue is going to protect. This dermal tissue is involved in the protection from the wind, stress, animals, etc. They all are coming under the mechanical stress, okay, or mechanical damage. So not only that, they are going to protect from the invasion of the parasites. Invasion means entry. Okay, so this tissue will prevent the entry of parasites into the plant body. Okay, this is another more aspect. And they are going to help in the protection from the disease-causing organisms. Disease-causing organisms like bacteria. Okay, from the bacteria or some kind of a fungus or whatever it may be, they are going to protect from the disease-causing microorganisms. Okay. Next is the dermal tissue is going to secrete a gum. Several layers above the epidermis forms a bark. You might have seen on the roadside. So, and remember one thing: if you have any doubts or questions, post it in the live chat section so that I can read and I can answer to your question or doubt. Okay. So next is what we have just discovered. They are going to secrete a gum. Okay, a gum is secreted. This gum is going to help in the repairing. So this gum is going to help in the repairing, and also the several layers of this tissue is going to help in the production of a bark. Okay, bark is nothing but in the plant body. So on the plant stem, on the roadsides you might have seen on the plant stem there is a bark. Okay, bark is nothing but a dead cells. So bark are nothing but, so they are dead cells. These dead cells are present on the bark. Okay, on the outer sides of the tree. Okay, so there are three types of dermal tissue. Okay, There are three types of the, again, it means again there is division, okay, division of cells. There are three types of the dermal tissue. What are the dermal tissue over here? So these dermal tissue are epidermis, okay, epidermis, epi means outside, remember epi means outside, so dermis means layer, means outside there is a layer, that layer is known as epidermis. So this epidermis is going to cover the, they are going to cover the cells, okay, these are these are a type of cells. These cells are going to cover the leaves, okay, stem, roots, etc. If you observe the leaf, okay, if you observe the leaf, so the leaf is covered, the leaf is slightly covered with a layer, okay, a layer of cells. This layer of thin layer of cells is known as epidermis. We will discuss the transverse section of leaf more in the 10th, okay. So what this epidermis is going to do? This epidermis is going to prevent the loss of water, and also it is going to prevent in the infection from the other parasites or infection from the other diseases, okay. So this is about it. And next thing is the stomatal guard cells. Stomatal guard cells are nothing but they are a small pores in the leaf. Let us consider this is the leaf. Underside of the leaf you will see small pores. Small pores or holes are there. These small pores or holes are known as stomata. So in the stomata there are a kidney shaped cells. They are known as guard cells. Okay, this is undersides of the leaf. Just now we have seen, no, these are undersides of the leaf. Okay, see, clearly observe it. If you clearly observe, there are cells. Okay, these cells are known as epidermis. Okay, so next one is these cells are going to have a stomatal guard cells. Okay, so the stomatal guard cells are nothing but stromata which containing guard cells. So see here, this is known as stomata. Okay, so this is known as stomata, and the stomata has a kidney shaped cells. These are known as guard cells. Okay, if you clearly see how the guard cells look like, these guard cells are going to look in this way. The green color portion in the center, okay, the kidney shaped cells. These kidney shaped cells are known as these cells are known as the guard cells. So guard cells are looking like kidney like structure, and they are present at the opening of the stomata. So if you go into the stomata, if you go even more into the through the stomata, through the guard cells, you will see a region that is known as stomata. So stomata looks in this way. So these are the cells, okay, and these are the guard cells. So these are the guard cells. So guard cells looks in this way. If you see from the longitudinal section, if you cut from the top to bottom, you can see in this way. So this is known as stomata. Okay, this is known as stomata. So these two are known as guard cells. These two are known as guard cells. How they are going to look? They are going to look like in this way. They are going to look like kidney shaped cells. Okay. So if the guard cells, what is the importance of the guard cells? They are going to involve in the closing and opening of the stomata. Okay, they involve in closing and opening of the stomata. Okay. So this is about stomata. Stomata means a pore which is present in the cell. Okay. So small pores. The guard cells are nothing but kidney shaped cells that are involved in, that are enclosing the stomata. Stomata are enclosed by the kidney shaped cells. Okay. And this is about it. And if you have any doubt or questions, okay, post it in the comment section. That is nothing but post it in the live chat section, so that we will discuss about your doubts or questions. Okay. So next is the root hairs. So the root hairs have the long hair-like structures. They are known as root hairs. So let us consider. So this is a plant. Okay. So this is a plant. So this is a ground. So the plant has a roots. Okay. This is the roots. So these are the roots and branches. So here if you see small fine hair-like structures, they are known as root hairs. If you closely observe, if you clearly observe under a microscope, you can see the root hairs are in this way. Okay, root hairs are in this way. So these are the cells. So we are observing the small cells over here. We are observing the small cell over here. So it is going to look in this way. So this is known as root hair. So this is very, very small. So if you observe, clearly observe, they look very, very tiny. They are going to involve in the absorption of water. Okay, absorption of water. So water will be taken by the root hair, and this water will be sent into the plant body for the photosynthesis, for the proper survival, and so on. Okay. So this is about root hairs. So next is stomata and root hairs of a dermal cells, dermal tissue are essential for the exchanging of the gases. It means they are involved in the exchanging of the gases. So how they are going to exchange? So exchanging is nothing but, so carbon dioxide, if you see here in this diagram, the carbon dioxide, so carbon dioxide is taken into the stomata, okay, and it will be taken for the cells. The cells is going to take the carbon dioxide, they will do the photosynthesis, and again oxygen will be produced. This oxygen is going to come out through the stomata. It means, can you see, there is exchanging of the gases. What gases are being exchanged? Oxygen and carbon dioxide are being exchanged in the stomata. Okay, so this is about root hairs. Okay, this is about stomata. So not only stomata, root hairs also going to exchange. They are also exchanging in the gases. We will discuss how the root hairs are being involved in the exchanging of the gases in your next academic year, that is in the 10th class. In the 10th class, we will clearly discuss about how the root hairs or roots are involved in the exchanging of the gases. We will discuss even clearly. Okay, but remember now, stomata and root hairs are involved in the exchanging of the gases. Okay, so they are made up of dermal tissue. Okay, so this is about it. And the next topic is the ground tissue. So what is meant by ground tissue? Ground tissue is a bulk portion of the plant body. Bulk portion of the plant body, it is made up of it means bulk portion of the plant body is made up of the ground tissue. So this ground tissue is mainly used for the storing of the food material. What is the food material? Example C6, okay, H12O6. It is a chemical formula for the glucose. Okay, so the glucose will be stored, and next water will be stored, and so on. It means these different products like glucose, vitamins, fats, proteins, etc. These all are stored in the ground tissue. What we are saying, they are the bulk portion. So bulk portion means the tissue which it is going to store something, that is known as a bulk portion. Okay, so here is it is useful for the storing food material and also physical support. Not only storing of the food material, it is also going to provide physical support. Physical support means, so it can withstand against wind. Okay, next it can withstand against rain, etc. Okay, so there are three types of ground tissue are there. They are parenchyma is one type, second type is a collenchyma. Okay, next one is a sclerenchyma. So these are the three types of the ground tissue. It means ground tissue can again divided into parenchyma, collenchyma, sclerenchyma. Okay, so before we discuss about before we enter into the next topic, if you have any doubts, let me know in the comment section. If you don't have any doubts, then also let me know that you have no doubt. Okay, so type it in the live chat section so that I will get one clarity that you don't have any doubts. Okay, so this is the reason I am asking you to mention whether you have doubts or no doubts. Okay, use the comment section, use the live chat section, so to ask or to let me know something. Okay, so next, so parenchyma. So parenchyma means these are the cells. These are the soft and also loosely packed cells based upon the function. Parenchyma. This is not parent. This is parenchyma. Okay, this is parenchyma. Okay, so the parenchyma is divided into chlorenchyma, aerenchyma, and also storage parenchyma. So these are the three divisions of the parenchyma cells. Okay, so next is, so chlorenchyma. What is the importance of the chlorenchyma? Chlorenchyma is a type of the parenchyma. So the word, look at the word. The word itself is saying about it. Chloren means it is about the chlorophyll. So it is a description about the chlorophyll. So chlorophyll is present. Okay, so chlorophyll is present in this tissue, that is known as chlorenchyma. Okay, so if you carefully observe, so in this tissue they are green in color. So why this chlorenchyma is green in color? Why? Because these cells are looking in green color. Okay, due to the presence of chlorophyll. So the chlorophyll, we know that the chlorophyll is green in color. Okay, the chlorophyll is which color? The chlorophyll is green in color. So why the chlorophyll is green in color? So it is why? Because it is a pigment It is a pigment. It is green colored pigment, so it is known as chlorophyll. So where it is present? It is present in the chloroplast. Okay. So what is the difference between chlorophyll and chloroplast? Chlorophyll, it is a pigment, and chloroplast is a organelle. Okay, chlorophyll is a pigment, chloroplast is the organelle. So this is only the difference. Okay, so this is about it, and the next one we will go for the parenchyma. So look at the word. The word itself is saying about it, what it is going to store. So chlorenchyma means it is going to store the chlorophyll, and parenchyma means it is going to store the air. Okay, parenchyma means air. So it is a type of parenchyma cells which have the air cavities or spaces. It means it is going to store the air. It is going to store the air. So that is known as parenchyma. Okay. So in this diagram, if you clearly observe under the microscope, under the microscope you can see the cells with the cells which are larger. Why these cells are larger? These cells are larger. Why? Because they have stored air. The air is stored in these cells, so they are parenchyma cells. Okay. So next is storage parenchyma. So what is storage parenchyma? Again, look at the same word. Storage means what? They are going to store. They are going to store something. So if the parenchyma cells are storing some amount of food material, that is known as storage parenchyma. So you can define as these are the parenchyma cells that stores water or food or waste products. If these components are stored, that cells is known as the type of parenchyma cells will become the storage parenchyma. Okay. So this is about storage parenchyma. So next is after parenchyma, next thing is a collenchyma. Okay, next one is a collenchyma. Collenchyma is they are thick walled and longer in size when compared to the parenchyma cells. So when you compare with parenchyma cells, these collenchyma cells have the thick cell wall. Okay, thick cell wall. Next is they are longer in size. Okay, so these are living and also provides structural support. Okay, if you observe under the microscope, you can see they are going to look like stones. Okay, that is collenchyma. So next one is a sclerenchyma. Sclerenchyma, these cells have the thick wall and also tightly packed without spaces. Means if you can see between the cells any spaces? No spaces are present between the cells. So they are known as sclerenchyma cells. Scleren, okay, chyma. So no spaces, thick walled cells are known as sclerenchyma cells. Okay. So next is the observation of types of parenchyma cells. So go to the laboratory. In the laboratory, take the slides of a parenchyma, different types of parenchyma cells, and observe. This is activity 9. So here observe the permanent slides. Observe the permanent slides of parenchyma, collenchyma, sclerenchyma that we have just discussed. Okay. So if you observe under the microscope, you can see these three types of the tissue. These three types of tissue can be observed under the microscope. Okay, and this is about our discussion about the plant tissue. In the next class, we are going to discuss about the next topic. And before we stop the class or before we end the class, if you have any doubts, let me know in the live chat section so that I can answer to your question or I can answer to your doubt. Okay, so let me know about your questions or doubts. Okay, if you don't have the doubts also, then also let me know that you don't have any doubts. Okay. So okay, then we will discuss the next topic tomorrow. Okay.